Hi everyone. In last 12 videos we have completed core fundamental understanding on dissolution testing. Now onward we will have 10 videos on dissolution method development for OSD formulations. In this video I will introduce dissolution method development as a structured science driven process for immediate release tablets. For this we will focus on solubility, method robustness, and discrimination. Let's get started. Dissolution testing is multifunctional. It is used for exploration, confirmation, and routine monitoring. Because these goals are different, the acceptability, variability, method flexibility, and sensitivity also differ from stage to stage. So even though the test looks the same, the purpose changes, and that drives different expectations. Method development should follow a structured approach. First, define the purpose. For example, QC release versus biorelevant understanding. Then understand API properties. For example, solubility, pK, polymorph, and particle size. Then assess formulation characteristic and then optimize hydrodynamics. For example, apparatus and rotation speed. And also select an appropriate medium and sampling points. This structure ensures the dissolution method is fit for the purpose, not just compliant. So first step is to characterize API. Actually, API properties drive dissolution behavior, such as weak acids dissolve faster at higher pH, while weak bases dissolve faster at lower pH. And for BCS class 2 and class 4 drug, stronger media or optimized hydrodynamics are often required to achieve meaningful dissolution. Solid-state properties such as polymorph and particle size directly affect reproducibility. That's why API characteristics guide the entire method, especially medium selection and rotation speed. Second step is understand the formulation. Fact is, formulation excipients strongly influence dissolution, such as lubricants can slow batting, disintegrants promote tablet breakup, and binders control mechanical strength. Together, these factors determine whether dissolution method needs low or moderate agitation. In short, formulation design controls how fast the tablet disintegrates and starts releasing the drug. Third step is to select apparatus. For this, it is important that the hydrodynamics must be stable and representative for reliable dissolution results. Apparatus 2, the paddle, provides uniform flow and the standard choice for most immediate release tablets. However, if tablet float or fail to stay immersed, the basket apparatus is preferred to ensure consistent exposure to the medium. Step 4 is to select dissolution medium. Method development start with initial solubility screening. For poorly soluble drugs, surfactants like SLS or twin may be needed, but they must be scientifically justified. Using too much surfactant can mask formulation differences and reduce method discrimination. The dissolution medium should balance adequate solubility with physiological relevance. Next step is agitation speed. So rotation speed or RPM directly affects dissolution behavior. 50 RPM is commonly used for more sensitive formulations, while 75 RPM improves method robustness and consistency. For hydrophobic tablets, higher agitation may be needed to prevent sticking or coning. However, RPM should never be so high that it masks real formulation differences. In short, RPM selection balances hydrodynamic stability with discriminatory power. Next step is to select sampling points. Sampling time points are critical in dissolution testing. Early time points capture the disintegration and initial release phase. They should be selected based on drug release mechanism and regulatory requirements, especially for F2 similarity profiles, which need several early time point samples. For immediate release tablet, early sampling is essential to accurately capture rapid drug release. Next is to evaluate discrimination power of the method. Actually, discrimination power is a key regulatory expectation for dissolution methods. This means a dissolution method should be able to detect meaningful formulation or process changes. For example, by differentiating between high and low hardness tablets or coarse versus fine granules. If the method cannot detect these changes, it is not clinically or scientifically relevant. In short, a good dissolution method must reveal real differences, not hide them. Next step is to validate method. For this, robustness testing is essential for a reliable dissolution method. This includes deliberately varying RPM, pH, media preparation, and degassing conditions to confirm method remains stable. Robustness method reduce OS risk and strengthen regulatory confidence in filings. Once optimized, the dissolution method must be fully validated before it is used for routine QC testing. So, dissolution method development is a systematic process, not trial and error. We start by defining the purpose, then understand the API and formulation, selecting the right apparatus, medium, RPM, and sampling points, and finally ensuring discrimination and robustness. This stepwise workflow ensures the method is scientifically sound, robust, and fully aligned with regulatory expectations. To summarize, for immediate release tablets, the dissolution method must be fast, discriminatory, and robust. Every step must be scientifically justified and properly documented. This method then becomes the foundation for QC release testing and stability programs. Thank you for watching. Please subscribe us for getting update on such industry application oriented videos and stay tuned because in this 70 video series on dissolution science, we'll cover dissolution method development, validation, troubleshooting, and many real industry example. In the next video, we will focus on dissolution method development for ER tablets. Post your question in comment section. I will respond it over there. See you in next video. Thanks.